Welcome back, everyone. In this module, we're going to zoom in on how evolution works and how it's written into our bodies at the genetic level. This is really the foundation for biological anthropology and that particular side of this course. Today's lecture is divided into two mini lectures. In this first one, we'll look at how scientists developed the theory of evolution by natural selection. We'll also meet Darwin and Wallace and unpack what natural selection actually says. Then we'll dive into Mendel's pea plants to see how traits are inherited across generations. In the second mini lecture, we'll move on to look inside the cell, into DNA, chromosomes, and cell division, and then out again to population genetics, speciation, and some contemporary issues like genetic testing. As we go, I want you to keep a core anthropological question in mind. I want you to ask yourself, how does understanding evolution help us think about human variation without sliding into racism, sexism, or other biological just so stories? So keep that in mind. The word evolution just means change over time. That's the broadest definition. Biological anthropologists use a more specific version. They talk about evolution as the process of change in the heritable characteristics of biological populations over successive generations. So notice what's not in that definition. Individual people, quote unquote, evolving superpowers, or evolution as a ladder from primitive to advanced. Evolution is about populations, genes, and time. It's not about individuals becoming better. For biological anthropology, evolution is the organizing framework. It helps us explain why humans share so much with other primates, why there's variation in traits like skin color, lactose intolerance, or disease resistance, and how our species emerged from earlier hominin species over millions of years. So when we talk about human biology and variation in this course, we are always implicitly asking what evolutionary processes produced this pattern. Darwin didn't invent the idea that species might change. Earlier thinkers were already pushing on this question. Think about, for example, what your textbook talks about as catastrophism. That's a framework associated with Cuvier, and it argues that Earth's geology and its fossils are the result of sudden catastrophic events, like global floods or massive disasters. Species in this framework were seen as being wiped out and then gradually replaced. Uniformitarianism, on the other hand, which was developed by Hutton and popularized by Lyell, argued that the same slow processes that we see today, like erosion, volcanic activity, and plate movement, have been shaping the Earth across all of deep time. That gave evolution the timescale it really needed to get going. Jean-Baptiste Lamarck offered an early mechanism and an early idea about species transformation. And his idea is the inheritance of acquired characteristics. So think about it like this. Stretch your neck for leaves, and your offspring are going to inherit longer necks. It's wrong in a strict genetic sense, but Lamarck was really important because he recognized organism-environment interaction, and he proposed that species could transform. The big shift with Darwin and Wallace is that they put all of this together with a clear mechanism, natural selection, which is grounded in evidence from nature. Darwin and Wallace both spent years doing field work in the tropics. Darwin was on the HMS Beagle, Wallace was in the Amazon, and then in Southeast Asia. And they noticed patterns like the famous Galapagos finches with different beak shapes that were suited to different foods. From their observations and from reading economist Thomas Malthus, they distilled three principles. The first is that traits are inherited from parents to offspring. Second, more offspring are produced than can survive. Resources are limited, so not everybody makes it. Third, offspring vary in their traits, and some of that variation is heritable. So if you put these three principles together, you get natural selection. Individuals whose traits give them an advantage in a particular environment are more likely to survive and reproduce, and their traits become more common in the population over generations. Darwin called this descent with modification. But a key anthropological reminder here: selection acts on individuals, but evolution happens in populations. Individuals themselves don't evolve in their lifetime. The modern theory of evolution didn't come from one person, didn't just come from Darwin. It was built from multiple scientific disciplines. So taxonomy and systematics, for example. These disciplines give us a way to name species and start to map their relationships. Linnaeus, for example, gave us the binomial system, Homo sapiens. And later scientists developed phylogenetic trees to show how species are related. Then geology and paleontology, they provide evidence for Earth's age and for the fossil record, showing extinctions, transitions, and massive radiations of life. Evolutionary biology and demography bring in mathematical and experimental tools that connect natural selection to population level change, which allows us to reproduce Darwin's insights using genetics. For anthropology, these disciplines underpin everything from how we place humans in the primate order to how we date fossils and how we reconstruct hominin family trees. While Darwin was thinking in terms of traits and populations, Mendel was growing pea plants in a monastery garden, trying to understand how traits are passed down. He worked with discontinuous traits, either purple or white flowers, round or wrinkled seeds, rather than continuous traits that exist on a spectrum like height. Mendel's key insights were that traits are controlled by discrete units, which we now call genes, and that different versions of a gene are alleles. For flower color, one allele might code for purple and another for white. Some alleles are dominant. Their effect shows up if you have even one copy, while other alleles are recessive, which means that you need two copies to see their effect. We use three terms a lot, and so I've put them on the slide here. The first is genotype. That's the alleles that you have. For example, two uppercase P's, an uppercase and a lowercase p, or two lowercase p's. The phenotype is what you look like. So that's a purple or a white flower. Homozygous versus heterozygous, having the same alleles versus different alleles. Anthropologically, Mendel's work is crucial because it gives us a clear way to think about inheritance and variation, and these are really the raw materials on which natural selection acts. Mendel summarized his findings in two principles that you'll see time and time again. First is the principle of segregation. Each individual has two alleles for a gene, but only one goes into each gamete, egg or sperm, and this allocation is random. Second, the principle of independent assortment. Alleles for different genes are distributed into gametes independently. So getting one allele for seed color doesn't force you to get a particular allele for seed shape. In the classic dihybrid cross, Mendel saw a 9-3-3-1 pattern in the F2 generation, which showed that traits weren't all stuck together, but were actually reshuffled. In humans, inheritance is often more complex. Many traits are polygenic, influenced by many genes and shaped by environment. But Mendel's logic still forms the backbone of how we think about genes and how we predict inheritance patterns, including in medical genetics and in forensic anthropology. Before we move on, I want you to pause and think about how ideas from this mini lecture show up in everyday life. So when I'm done talking, pause the video and take a few minutes to respond in a notebook, a document, or wherever you're taking your course notes to a few questions. The first question is, where have you heard people talk about evolution or survival of the fittest? This could be in a biology class or on social media, in a movie or a TV show, in conversations about politics, race, or human nature. Jot down just one or two concrete examples. Then I want you to ask yourself, what do they seem to mean by it in that context? Are they using evolution to mean improvement, progress, getting tougher, or something else? Are they talking about individuals changing or populations over time? Finally, how does that compare to what you just learned about evolution and inheritance? Think about Darwin and Wallace's natural selection, and think about Mendel's ideas about genes and alleles. In your examples, are people using the term in a way that fits the scientific meaning, or are there misunderstandings? Spend a few minutes with this, and when you're done, unpause the video and we'll zoom in on DNA, cells, and how all of this works at the molecular level. Now that you've reflected, we're going to zoom in from traits and populations to the cell. But remember, evolution changes allele frequencies in a population, but alleles are carried on chromosomes inside cells. In humans, who are eukaryotes, our cells have a nucleus that houses DNA, mitochondria that produce energy and that contain their own small set of genes are in there as well as ribosomes that build proteins and other organelles like the endoplasmic reticulum and Golgi body, which processes and ships molecules. We also distinguish between somatic cells and gametes. Somatic cells are most of your body cells. In these cells, they divide by mitosis, which we'll go over in a little bit more detail later, and they're diploid with 46 chromosomes. Gametes are eggs and sperm. They are haploid with 23 chromosomes and are produced by meiosis. When egg and sperm meet, they form a zygote with a full set of 46 chromosomes. And that's where a new combination of alleles enters the world. At the molecular level, heredity is written in DNA, deoxyribonucleic acid. Most of it sits in the nucleus as nuclear DNA, with a smaller amount in the mitochondria as mtDNA. DNA itself is made of nucleotides, each with a sugar, a phosphate group, and one of 4 bases, either A, T, C, or G, which stands for adenine, thymine, cytosine, and guanine. The famous double helix structure is sort of like a twisted ladder. Base pairs, A with T or C with G, are the rungs. The sugar phosphate backbones are the sides. And during replication, the strands separate, and each serves as its own template to build a new complementary strand. Then RNA, or ribonucleic acid, it's similar, but it uses U, or uracil, instead of T. and it's usually single-stranded and plays a really key role in protein synthesis. DNA is transcribed into RNA, and RNA is translated into proteins. Sequences of 3 bases, or codons, specify amino acids, which are then strung together to make proteins, everything from enzymes to structural proteins. And that is essentially how a tiny sequence difference can change something as big as disease risk or lactose intolerance. Mitosis and meiosis are the 2 main ways that cells divide. In mitosis, a somatic cell makes an exact copy of itself. 46 chromosomes duplicate and then are evenly split into 2 new diploid cells. This keeps tissues growing and repairing. In meiosis, a germ cell goes through 2 rounds of division. 1 diploid cell becomes 4 haploid gametes with 23 chromosomes. Here we get crossing over and random assortment, which mix parental alleles and create new combinations. Sometimes, however, the process misfires, and that's non-disjunction, when chromosomes fail to separate properly. It can lead to all sorts of conditions like Klinefelter syndrome or Turner syndrome, among others that your textbook mentions. For anthropologists, understanding these processes helps us talk about human variation and health in a way that's scientifically accurate and sensitive. What we don't want to do is turn people's bodies into deviations from some sort of imaginary norm. In population genetics, we define evolution more precisely as a change in allele frequencies over time. Your textbook highlights 4 main mechanisms that change allele frequencies. The first is natural selection. Differences in reproductive success based on heritable traits, and it produces adaptations. Then there's mutation. Random changes in DNA that create new alleles, most of which are neutral, some of which can be harmful, and a few are beneficial. Then there's genetic drift. These are random changes that are especially powerful in small populations. The genetic drift includes things like bottlenecks or drastic reductions in population size, as well as founder effects, when a small group leaves to start a new population. Finally, there's gene flow, which is the movement of alleles between populations through migration and interbreeding. Gene flow tends to increase variation within populations and reduce differences between populations. A key anthropological takeaway here is that human populations have experienced extensive gene flow for a very long time. That's one reason that there's only one human species, and it's why simple racial categories like black and white do a really poor job of capturing genetic variation. When does evolution produce a new species? Biologists often define a species as a group whose members can produce fertile offspring with each other. There are some exceptions, but this is sort of a useful starting point. 2 key comparative concepts are really useful for understanding processes like speciation, and those are homology and analogy. Homologous structures are similar because of shared ancestry. For example, the bone pattern in a human arm, a cat leg, a seal flipper, and a bat wing, like you have pictured on the slide in front of you. Analogous structures are similar not because of shared ancestry, but because of similar selective pressures. For example, the wings of birds, bats, and insects. Speciation, or the splitting of one lineage into 2, can happen via allopatric speciation, where populations are physically separated and diverge, or sympatric speciation, where reproductive isolation evolves within the same geographic area. Over time, we can see adaptive radiation. One ancestral species spreads into new niches and diversifies into many species with different adaptations. You can think of, for example, Darwin's finches or Hawaiian honeycreepers. The chapter then jumps forward to the biotechnology and the genomics era. Biotechnology uses artificial methods to modify genetic material, developing vaccines, genetically modified crops, or potential gene therapies. Genomics looks at entire genomes. It works on things like mapping genes, comparing species, and studying how coding and non-coding regions interact. For humans, this includes genome-wide association studies, or GWAS, that link genes to complex traits as well as diseases, as well as to analyses showing that humans and chimpanzees share about, this is a big number, 98.8% of their DNA sequence. Then there are things like direct-to-consumer testing. Think about companies like 23andMe or Ancestry DNA. There are many others. These consumer testing companies are interesting. On the plus side, they're useful because people can learn about carrier status for certain medical conditions, or they can explore ancestry in new ways. But there are downsides as well. Tests can be misunderstood. They can create a lot of anxiety. They can reinforce pretty shaky ideas about race and identity. And they raise privacy concerns, especially as law enforcement starts using these databases. For anthropologists, this is a really rich site for studying how people interpret genetics, identity, and risk, and for asking who benefits and who may be harmed by the genomics industry. The final section of the chapter I had you read for this module steps back and asks, how do science and culture collide around evolution? A few key clarifications here. In science, a theory is not just a guess. It's a framework that's supported by massive, massive evidence, like the theory of gravity or atomic theory. Evolution is a theory in this sense. Second, evolution does not explain the origin of life itself. It explains how life changes and diversifies once it exists. Finally, evolution is not goal-directed. There is no pinnacle species or some inevitable march toward humans. No. Evolution is more like a branching tree, or even something like tangled roots. It's more like those than it is an escalator. The chapter you read also reviews legal battles in the US over teaching evolution in schools, from the Scopes trial in 1925 to more recent attempts to introduce creation science, or intelligence design, into science curricula. Courts have repeatedly ruled that these are religious views, not science. For anthropology students, this history matters because it shows how ideas about human origins are never just neutral facts. They're really deeply entangled with politics, religion, racism, and national identity. And part of our job is to sort out how empirical claims are distinct from value claims, and to be really honest about how our own beliefs intersect with scientific evidence. Before we wrap up, I'd like you to pause the video and respond to a short reflection prompt. I want you to think of one way evolutionary thinking shows up in public debates today. They could be debates about health, race, gender, human nature, you name it. How is it being used well? And how is it being misused? Then I also want you to think about how those understandings of evolution change populations. Sorry, small technical mishap there. Anyway, the second reflection prompt that I want to throw out there for all of you is, how does understanding that evolution changes populations, not individuals, shift the way that you think about human difference? We'll build on these reflections later in future modules, but I want you to take just a few minutes to think about them now. When you're ready, unpause the video, and we'll move on to our last slide. All right, to wrap up this module, I want to lay out just a few key points to summarize. First, evolution is best understood as change in allele frequencies in populations across generations, and it's driven by mechanisms like natural selection, mutation, genetic drift, and gene flow. Then Mendelian genetics, molecular genetics, and population genetics all give us tools. They help us connect genes in cells to patterns in populations, and eventually to the emergence of new species. Finally, the science of evolution is really robust, but the stories that people tell about evolution, especially about humans, are deeply, deeply shaped by culture, politics, and history. In the next module, we'll shift from evolution in general to human biological variation more specifically, and we'll talk about how anthropology tackles race, genetics, and environment. Thanks very much, everybody.